Good afternoon, everyone. I would like to introduce the speaker for today, Dr. Sailu Elleboina, talking about Sir's employment and education. Sir is currently working as an associate professor at University of Hyderabad, Central University. Discipline is in bioinformatics and systems biology. Sir has done his postdoctoral fellowship during the period August 2009 to June 2011 at National Institute of Health, Research Triangle Park, USA, in bioinformatics and systems biology of embryonic stem cells. The project was integrating genomic datasets to identify the novel regulators of embryonic stem cells. He has worked as project associate at CBIT-Hyderabad during the period August 2005 to July 2006. He's obtained his PhD in computational genomics from CBIT-Hyderabad in the year 2005, and his work included in silico prediction of regulons in bacterial genomes. Master of Science from University of Hyderabad in biochemistry, bachelor's in science in chemistry and biology from Kakatiya University. Well, there are many grants that were sanctioned under Dr. Sailu's name. The first one including integrated genomic database of stem cells. This was sanctioned by DST for 23 lakhs during the period 14 to 17. And another one would be integrative approach for identifying host pathogen interactions, funded by NIAB for 10 lakhs in the year 2014. Well, Sir has submitted several projects, including one approved by DBT, University of Hyderabad. This was on development and application of high-resolution genome conformation capture technology to investigate genome architecture in space and time. And another one was for 13 lakhs. This was on integrating diverse genomic data sets to identify potential bioterrorism signatures in genome sequences. Sir has received many awards and honors, the first one being travel grant awarded by the Department of Biotechnology, India in the year 2003, visiting fellowship by National Institute of Health, USA, for the period 2006 to 8 and 2009 to 11, and the Best Poster Award in the Conference on Genome Architecture and Cell Fate Regulation, December 2014, organized by University of Hyderabad. Well, the list goes on actually, and talking about publications, Sir has about 15 publications in total and 4 conference proceedings, and Sir has vast mentoring experience for JRFs, SRFs and postdoctoral fellows, and he's guided many MTech and MSc theses as well. So it's a great honor and a rare opportunity that we get to listen to such eminent speaker. So thank you very much for accepting our invitation, and I request you to begin the session, sir. Thank you.
Thank you, Sumitra, for kind introduction and invitation. This is the outline of my talk: genetic polymorphism and flow of genetic information, point mutations and SNPs, annotation of SNPs, toxicogenetics. Generally, in biology, polymorphism is the occurrence of 2 or more morphs and or phenotypes in populations of species. In order to identify the polymorphism, the morph must occupy the same habitat and they should belong to the randomly mating population. Polymorphism takes place when there are 2 or more possibilities of a trait on a single gene. This is an example of morphological differences due to changes in metabolite. The people with the black skin will have a eumelanin, and the people with the white skin will have a pheomelanin. This is another example of morphological differences due to changes in metabolite. This individual has high eumelanin levels. This individual has high phenylalanine levels. These are the morphological differences due to changes in protein structure. The individual with the curly hair will have a different sequence and structure of tricholine protein. This individual has a different sequence and structure of the tricholine protein. In order to understand the differences in phenotypes, you must understand the differences in functional elements. If you look at the functional elements in the eukaryote genome, these are the gene structure and these are the structure of regulatory elements. The gene will have 5' UTR, exons, introns, and 3' UTR. The regulatory elements will have promoters, proximal promoter elements, and distal regulatory elements, which consist of insulators, silencers, and enhancers. If you look at the flow of genetic information from gene to phenotype, this is the genome and functional elements, and the changes in these functional elements reflect the changes in transcriptome, and changes in transcriptome reflect the changes in proteins, and therefore finally that reflects changes in metabolites. So here the phenotype can come through the differences in differences in metabolites, and also it can come through at the protein level due to structural changes in the protein, and it can also arise due to changes in non-coding RNA genes. This is the flow of genetic information from the genome to phenome. This is the genome, transcriptome, proteome, and metabolome and phenome. And again, phenome can arise due to differences in metabolome or differences in proteome or differences in non-coding RNA genes. Polymorphism can be differences in any measurable phenotype. It can be variations in response to the exposure, such as alcohol or drugs, variations in response to the treatment, variations in particular genetic disease phenotype, or variations in resistance or susceptibility to infection. It can also be differences in the molecular phenotypes or levels of metabolites. It can be differences in melanin, and it can be differences in blood sugar levels. It can be differences in cholesterol levels. It can also be differences in metabolites or toxic compounds levels when the person is ingested. So finally, the variations in genes are responsible for the variations in morphological and molecular phenotypes. All the variations in morphological differences arise due to mutations and mostly due to point mutations. A point mutation or a substitution is a genetic mutation where a single nucleotide base is changed, inserted, deleted from a sequence of DNA and RNA. There is a change in single nucleotide base, or there is an insertion and deletion of single nucleotide base. If you look at the point mutations, so it can be base indel, deletion or insertion of a single base, or a base change. These changes can be anywhere in the genome. It can be there in the coding genes, non-coding genes, and regulatory sequences. So if you look at the point mutations in coding genes, the base indels will have a frame shift mutations. It will entirely change the protein sequence of the amino acid sequence of the protein. That will result in drastic change in the function. The base change can be synonymous mutation, which result in the degenerate codon. It can be non-synonymous mutation. The non-synonymous mutation, again, it can be missense mutation and nonsense mutation. The missense mutation can be conservative and non-conservative. I will give you detailed explanation of these terminology in the next slide. So if you look at the point mutations, so this is the codons in the reference individual or normal individual. If you look at the second individual, the mutation changes to another codon, but the codon still codes for the same amino acid. This is called synonymous substitution. It results in synonymous codon. If you look at the third individual, The mutation leads to stop codon, which results in the premature or truncated protein product, which will have a drastic effect on the protein function. If you look at the missense mutation, it can be conservative and non-conservative. So in conservative missense mutation, there is a different codon. There is also a different amino acid, but the amino acid will have a still similar physicochemical properties as a original amino acid. You can look at here. This is acidic, and aspartate is also acidic. And if you look at the last individual, the missense mutation leads to different non-conservative substitution. So this is a lysine. If you look at the physicochemical properties of the lysine and glutamate and aspartate, lysine is entirely have a different physicochemical properties. You can see this is a amino terminal with basic properties, and these aspartate and glutamate has the acidic properties. The genetic polymorphism is defined as the inheritance of trait controlled by a single genetic locus with two alleles, in which the least common allele has a frequency of about 1% or greater, and the population should be randomly mating. So if you look at the single nucleotide polymorphism or SNP, a base change mutation that is present in more than 1% in a random set of individuals in a population. SNPs can be found in anywhere in the genome, coding or non-coding regions. So we have to consider random set of individuals, because if we take particular population from particular region or particular ethnic category, they will all have a similar sequences. So the SNPs belongs to particularly that population. If you want to have SNPs of Indian population, you need to randomly select the population from different ethnic regions and people speaking from different languages. So these are the different individuals. This is a toy example of chromosome 2. It has a copy 1 and copy number 2. So one is coming from mother and another one coming from father. It has an allele. Here it is homozygous. This is heterozygous allele. And again this is heterozygous. And this is homozygous. If you look at the chromosomes, each chromosome is a double-stranded DNA. So this is same representing as a double-stranded DNA. This is coming from mother and this is coming from father. And if you look at the sequences, this is a double-stranded DNA, this is another double-stranded DNA coming from father. If you look at the variations, there is only the variation in single base. So this is a point mutation. And if you look at the number of alleles, there is a TA allele, one allele, this is homozygous, TACG, this is heterozygous. So the individuals have an allele TA and another allele CG. So if you look at the allele frequency, the TA allele is 83% and CG allele is 17%. So TA allele 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 out of 12 is 83%. The CG allele frequency is 2 out of 12, 17%. So the minor allele, which is second most frequent allele, is called minor allele. The CG allele is minor allele, and the TA allele, which is most frequent allele, is called major allele. In order to identify the impact of SNPs and protein function, in order to identify the mechanism of morphological phenotypes, you must annotate the SNPs in genome sequences. So as I told you earlier, the genome sequence can have genic regions and regulatory regions. Again, the regulatory regions can have insulators, silencers, enhancers, and promoter sequences. So this is a classification of the genic regions. If you take any DNA, it will have a genic region, non-genic region. Again, genic region will have a coding genes, non-coding genes, and coding regions will have exons, splicing regions, introns, and UTRs. The regulatory regions will have promoters, enhancers, silencers, and insulators. So if you look at the mutations, these regions, if there is a mutation, will have a high impact on the function. Therefore, it is likely to be more lethal. And this is less likely to be lethal. The mutations in regulatory regions are less likely to be lethal. So if you have to annotate the SNPs, you need to identify where the SNPs are falling. Which part of the coding region, which part of the regulatory regions. For example, this is a coding region. So this SNP is falling in the coding region. And again, within the coding region, it can be synonymous SNP, non-synonymous SNP. And again, non-synonymous SNPs can be nonsense SNPs or missense SNPs. And missense SNPs can be conservative and non-conservative. So if you look at this region, here the SNPs are falling in the promoter region and the promoter or regulatory regions. And these mutations in these promoter regions are likely to result in change in the expression level of the genes. It doesn't lead to change in the coding sequence, but it can influence the expression level of the neighboring genes. They are called ESNPs or expression SNPs. We also call these loci as a expression quantitative trait loci. If the trait is quantitative trait, we call this as a expression quantitative trait loci. So the ESNPs can be in anywhere in the regulatory elements, promoters, enhancers, silencers, insulators. They are mostly located either upstream or downstream of the gene. So these are the different types of SNPs. SNPs can be in regulatory regions. They are called ESNPs. SNPs in the coding regions, again, can be synonymous SNP, non-synonymous SNP, missense SNP, and nonsense SNP. Again, missense SNP can be into two types: conservative SNP, which cannot which will have a least impact on the function; non-conservative SNP, which will have a high impact on the function. So genome sequence functional elements can be categorized into 52 different terminologies. So that is called sequence ontology. If any of the SNPs fall within these regions, we classify them as this particular categories. If you see the mutations or variations in chromosomal number variation or exon loss variant, they are likely to have a high impact on the function. Stop loss, loss of the stop codon, start loss, loss of the start codon, stop gained, which is a nonsense mutation. It's a gain of the stop codon. So if you look at these sequences, variations in these sequences will have a moderate impact on the function, whereas the variations in these sequences will have a low impact, and variations in these sequences will have a very, very low impact on the function. And these sequences mostly belongs to the non-coding regions. This is an example where we have sequenced different individuals and identified SNPs in the different sequences, sequence regions in the genome. If you look at the SNPs in introns and intergenic regions, they are very high in comparison to the splice site donor, because the mutations in introns or intergenic are least likely to have an effect on the function compared to the splice site donor. That's why they are abundant across the different individuals. Similarly, distribution of indels also, they are high in the intron and intergenic regions and low in splice site regions. So if you look at the impact of SNPs on function, the SNPs with high impact regions, within the high impact regions, are very low in compared to the within the low impact regions. You can see You can see these SNPs, they are very high in regions with low impact. There are various applications of SNPs. Since SNPs are large number, they are distributed throughout genome and they are highly variable. Therefore, they are very good candidate markers for genome-wide association studies. Association studies can determine whether a genetic variant is associated with a disease or a trait or a particular trait. In the area of pharmacogenetic and toxicogenetic studies, SNPs can be used to adjust the dosage according to the individual genetic profiles. The SNPs can also be used for assessing biodiversity and phylogeny. There are various SNP databases, HapMap project, database of SNPs, and Ensemble variation database. Coming to the transitions and transversions. So transitions are conversion of one class of nitrogenous base to same class of nitrogenous base. So you can classify the nitrogenous bases into two classes according to the number of rings. Adenine and guanine will have 2 rings, whereas a cytosine and pyrimidine has a 1 ring. So here is a transition. Converting 2 ring to 2 ring is a transition. Converting to same class, 1 ring to 1 ring is also transition. Whereas the converting 1 ring to 2 ring or 2 ring to 1 ring is called transversions. So if you look at the number of possible transitions, there are 4, blue color, 1, 2, 3, 4. If you look at the number of transversions, possible transversions, there are 8. 1, 2, 3, 4, 5, 6, 7, 8. So therefore the expected ratio of transition to transversion is 4 by 8. It is 0.5 or 1:2. This is the observed ratio of transitions to transversion. So if you look at this percentage stacked bar plot, this shows the percentage distribution of different conversions across different individuals sequence. You can see adenine to guanine, both are purines, belongs to same class. This percentage is high. The conversion of thymine to cytosine, both are same class. Again, this percentage is very high. If you look at the third one, cytosine to thymine, same class. Again, this is high. Guanine to adenine conversion, this is also high. And if you look at the fifth class, cytosine to adenine, they belongs to different classes. All these 8 possible conversions, they are showing very low percentage abundance across the individuals. That means the observed ratio of transitions is much more than the observed number of transitions are much more than the expected number of transitions. Because the substitution of 2 base ring with another 2 base ring will have a least likely, least impact on the DNA sequence, DNA structure. If you substitute 2 ring with another 1 ring, it will have a much more impact. So that's why the observed transitions are more. If you look at the ratio, the observed ratio, it is coming approximately 2.1 or 2.3. The observed number of transitions are much more also due to degenerate codon. For example, if you look at the amino acids with 2 codons, if you look at glutamic acid, the OPUL base, the third codon, both are having same class of nitrogenous bases. Adenine and guanine, they both have 2 rings. If you look at the aspartic acid, cytosine and thymine, they both have 1 ring. And lysine, they both have 2 ring, adenine and guanine. So therefore, transitions are less likely to result in amino acid substitution because the verbal base contains the same class of nitrogenous base. These are the applications of the transitions and transversion ratio. It can be used for identifying the quality of the genome sequence, and it can also be used to filter the SNPs. Generally, in the humans, the observed transition to transversion ratio is 2.1. If it is less than 2.1, if it is drastically more than 2.1 across the genome, that means there is something problem with the genome sequence or SNP calling. The ratio can also be used as a quality indicator for single nucleotide polymorphisms inferred from high throughput sequencing data. The SNP ratio varies greatly by genomic region and functionality. So if you look at the human exomes, the ratio increases to 2.8 to 3.0 or higher, because the coding sequences, in the coding sequences, the transitions are less likely to have changed the protein sequence. Coming to toxicogenomics and toxicogenetics, the toxicogenomics combines toxicology and genomics with other high-throughput molecular profiling technologies such as transcriptomics, proteomics, and metabolomics. If you look at the pharmacogenetics or toxicogenetics, it identifies the genetic markers that predict differences in pharmacokinetic and pharmacodynamic properties or admit properties across the different individuals. So if you look at the individuals in the population, each individual will have a capability of metabolizing a drug or xenobiotic differently. Particular individual may metabolize fast, another individual may metabolize slow. These are all due to differences in the genes encoding for all these pathways. Phase 1 metabolism, phase 2 metabolism, and phase 3 metabolism. Phase 1 metabolism will mostly have a cytochrome genes, and variations in all of these pathway genes results in differences in admit properties. So in pharmacogenetics and toxicogenetics, these are some of the important terminologies: therapeutic index and therapeutic window. So if you look at this graph, this shows x-axis shows the dosage of the drug, and y-axis shows percentage of individuals responding to the dose. If at this dose level, 50% of the individual are responding to the drug, they will have a beneficial effect at this dose. So if you look at this one, toxic effect TD50, the dose at which 50% of the individual are showing toxicity, and LD50, lethal dose 50, at the dose at which 50% of the individuals are showing lethal effect. Generally, if you look at this therapeutic index LD50 by ED50, it is only considered for animals, because we don't do trials which kills the humans. And if you look at these humans, we calculate TD50 and ED50. A drug with high therapeutic index is a very good drug. And other terminology is a therapeutic window. The therapeutic window is the range of drug dosages which can treat disease effectively without having toxic effects. The range, so it should not have a toxic effect. So the range of the maximum range of the drug is here. The concentration should not exceed here. And the minimum dosage, ED50, the dosage at which 50% of the individuals are responding to the drug. So this range is called therapeutic window. This therapeutic window can be used for identifying whether it's a good drug or bad drug. So if you look at this drug A, this has a narrow therapeutic window. That means we really need to adjust the dosage according to the individual metabolic capabilities or individual genetic profiles of the genome-biotic metabolic genes. If you take drug B, it has a large therapeutic window. We really don't have to worry about dosage of the drug. It is effective at the 80-50. If you look at the high toxic dose, it is much more than the where it is showing your effect on the 100% of the population. So you don't have to worry about the doses here. But in this drug with a narrow therapeutic window, you really need to worry about adjusting the dosage. That's where you need to study about the genetic differences among the individuals. These are some of the drugs with narrow therapeutic window. Warfarin. This is an example, perfect example for adjusting the dosage. Some people with low dose of warfarin, even with low dose of warfarin, they will have a bleeding effect. And some people need high dose of warfarin to stop the clotting. There are other antibiotics also falls in the narrow therapeutic window. Vancomycin, gentamicin, amikamycin, and another important drug, valproic acid. And recently this hydroxychloroquine also, it has some toxic effect or cardiotoxicity and other toxic effects. So hydroxychloroquine, it will be better if you adjust the dosage according to the individual genetic profiles. Thank you.
Good evening, everyone. Well, now it's time to thank Dr. Sailu Ellaboi-Nagaru for delivering such an informative and interesting session. So thank you for accepting our invitation and being a part of this one-week webinar cum FDP series on current progress and future prospects of biotechnology. Thank you, sir. Your session was full of passion and zeal. And thank you very much for sharing your thoughts and experiences. It is indeed a great honor for us to have listened to you, sir, where you have shared your profound knowledge. Thank you very much, sir.